Well, I want to start with talking about your coming to the US and talking about how your interest in science developed.
Sure. Well, I am an immigrant. My family immigrated from Taiwan to the US when I was a kid. And I actually went straight to high school in upstate New York, which is an all white community. Obviously, that was a rude awakening for me. Speaking very little English, it took me quite a while to acclimate to the environment. But I think just like anything that once you get thrust into a challenging environment, and you rise to the occasions and survive, and I did. So sort of fast forward to my college days, I was fortunate enough to get into Harvard, and I think some of this I would attribute to affirmative actions. I mean, I know there's a lot of controversy about affirmative action. There's even a case pending in the Supreme Court, particularly about Harvard and involving sort of the Asian Americans application. So I would certainly think that I was a beneficiary of that actions, and I would not have been in where I am today without that support.
Were you interested in science in high school? Did you take that with you to college?
Yeah. I mean, I did. I was fortunately introduced to a faculty member at Rockefeller University in New York City, and so I spent sort of part of my junior year and senior year in summer and working there, and I would say that's what piqued my interest. All right, and so that continued during my college and medical school days. So when I was in college, I also expressed interest in working on various different scientific projects. I continued to work with my mentor at Rockefeller Universities during college, and we actually were very productive in publishing papers. So that's kind of continue, and so I certainly thought that was sort of my career path because that's where my interests lie. And I mean, I would say what I thought about other career path, I did think a little bit about it, but kind of unfortunately in a way that I would say maybe I was sort of the victim of the Asian stereotyping that I thought maybe science is the one that I could thrive, although I certainly would not have chosen otherwise.
So who was that mentor you spoke about at Rockefeller University?
Yeah, his name is Gerd Griniger. Actually, he came from Germany and came to the U.S. to study. So he certainly has a history of immigrations and how the U.S. benefit from people immigrated from the rest of the world.
So he gave you some help sort of fitting in as well as scientifically, I guess.
Yes, definitely. He was my first mentor and certainly set the standard for me. He certainly has a very high standard and kind of had imparted on my future style in terms of working.
Okay. When did hepatitis come into the scene for you?
Well, I would say that the initial work I did at Rockefeller was related to liver. All right, the liver, and so that sort of has always been my interest. I've always been intrigued by liver and what it does and how diverse that it carries physiological functions. So when I would say in maybe in medical school, I got exposed to virology. And so I became interested in virologies, and actually I work with my medical school mentor. His name is Tom Benjamin, who is a professor at Harvard Medical School. He works on viruses. So I kind of work with him. I really develop an interest in viruses and how does the virus interact with the hosts? How does virus infect the cells? So I really became that interested in that process. And then sort of moving forward, and when I decided to focus on certain area of research in my specialty, because I went into, after medical school, I did my residencies in internal medicines. From there, I focused on gastroenterology and hepatology. And so I was trying to find something that would be relevant to my previous experience. And so you can see that with liver and viruses, viral hepatitis became a natural sort of path for me. And also when I was doing my fellowship, there was a person there who is very prominent in viral hepatitis research and who's also a physician scientist. His name is Jack Wans. And I worked with him when I was a fellow. And then eventually I developed my own laboratories and becomes an independent investigator with faculty appointment as assistant professors for several years until I was recruited here to NIH and NIDDK.
Okay. So the internship was at Massachusetts General, right?
No, actually, the internship was at NYU Bellevue, because I decided to come down to New York for my residency, because my fiance then was going to law school in New York. So we kind of jointly decided to move down. And so I spent 3 years as a residency. Actually, it was great that I was able to get away from the Harvard environment, right? I've been there for 8 years, more than that. So it was good to sort of get away to kind of see what other places are like. And I think was very nice. And I also, because that's when AIDS sort of came out with the whole virology, right? So it sort of really intrigued my interest because of my interest in virology, although I didn't think that I would want to work on HIV, AIDS. I was thinking about, obviously, there are other viruses. So, but nevertheless, that my experience at NYU Bellevue really gave me a very clear view of what the virus can do to devastate a person's immune systems and causing disease like AIDS. So that really had a great impact on me. And I have taken a lot of patients with AIDS when I was there. And so I kind of really fully understand it. So then I decided we decided to move back to Boston for my fellowships. That's when I did my fellowships at MGH under Kurt Isselbacher, who was certainly a sort of a career mentor for me as well. And so that's kind of really set the stage, what I wanted to do. And then sort of the rest is history. I've been continued to working on viral hepatitis since my fellowship day as a faculty member at Harvard, and then here as a section and then branch chief.
How do you think that internship, that the time you spend in the hospital, how does that affect you as a scientist? That's something that certain PhDs don't necessarily have.
Yeah, I think that gave me a unparalleled perspective on how I see the science, health, and medicine can intersect, and how we can really leverage the knowledge we learn on both sides. Because I spent quite a few years in the laboratory before I actually had the chance to work in the clinic. So that really made me think, how can I do better? How can we do it better as a physician scientist? How can we translate what we learn, basic science, into clinical medicine? And that's always been my motto. I always thought of my research always been sort of at the interface of clinical and basic science. And I continue to do that. And I think for people like me, have that unique perspective in terms of what the patients or healthcare means and how we can make it better, and so that's really kind of a set kind of the stage for my career pursuit.
So now somewhere in here, in 1989, hepatitis C had been cloned and characterized. That must have been a pretty big turning point as far as looking at the science you wanted to do.
Yeah, certainly. I mean, for those of us who work on viral hepatitis, we have known of the existence of so-called non-A, non-B hepatitis for many years. And really, as you said, that with 1999, the groundbreaking discovery of the hepatitis C virus by Michael Halton and his colleague Chiron really opened the door for lot of research, and specifically trying to understand the virus, what it is, and how does it cause disease. So from that point of view, that really kind of break the whole field wide open. And for me, that was just at the right time, right? Because I was in 1889, I was just becoming a junior faculty member or finish my fellowship. So I quickly pivoted my research to looking at hepatitis C virus, although my interest has always lied in hepatitis B virus. That's where I did a lot of my work as a fellow, when I was a fellow. There's a personal reason for it because when I was a kid, I had acute hepatitis B, and some of my family member did. As Taiwan is an endemic for hepatitis B, right? So there was a personal connection for me to decide what to work on. So it's not just totally science. I do have a personal story about in that. So I've always interested in hepatitis B, and even up to today, I continue to work on hepatitis B, and there's still a lot to do. And so I would say my research sort of combine both hepatitis B and hepatitis C. They are very different viruses, but they both cause the same disease, right? So from that point of view, my clinical point of view, it makes sense, right? So we're looking at different pathogens, but causing the same disease of the organ that I'm interested in.
Right. I'm glad we got to that sort of other motive for you getting interested in hepatitis. That's fascinating. Mm-hmm. Some of your early work, I guess this was in your fellowship, was in your receptor tyrosine kinase experiment. Yeah. And I was really intrigued because you worked or got some help from George van der Woodau.
Sure, who's a main NIH, major NIH figures, right, for many years. Yeah. Well, I would say that's a little bit of my scientific detour. I kind of got interested in it because I read some interesting paper by him and how this fusion events that are causing this tyrosine kinase to be oncogenic, and there's some implications suggest that it has a relevance in terms of liver regenerations and malignant transformations, because liver is what I worked on. So I sort of a foray into this, collaborate with some folks, generate some animal model and looking at it, and it turned out actually they play a major role in breast cancer developments, and I kind of did a little bit of work on breast cancer. This is all during my fellowship and junior faculty level, and we published some nice papers. So I would say that once I moved here, because sort of my focus, I think I need to really build a program in where the NIDDK has a major interest in, right? Liver disease. Sure, liver cancer may be relevant, but it's really more of an NCI, although I continue to work a bit on liver cancer because of the viral hepatitis viruses. But I kind of pivot back to viral hepatitis. So I think that was a detour. I mean, that's science, right? I mean, you kind of go with where flow is, and we got some good publications out of it, and I even got some NIH funding on that. So, but nevertheless.
You went right to my next question, which I wanted to talk about you being a grantee, an NIDDK grantee. Yeah. And what your impression was of the institute in that period.
Well, I didn't know much about specific NIDDK. I do know about NIH, and I think my first grant was a CADE award, which is sort of for sort of transitional faculty member, right? So that's the CADE series. So that was my first grant, actually, and I would say that really set the stage for my ongoing scientific pursuit, that really allowed me opportunity to focus on what I want to do and give me protected time in an academic institution to pursue what I want to do. So I think that was really important rite of passage, and I certainly very appreciative. And I dealt with some of the program people who run the K Award, and they were very encouraging, very helpful, and very supportive. So from that point of view, I'm certainly very appreciative, because I know by getting that K Award, I have my foot in the door. Right? I mean, that's how important it is. And I was able to get a couple grants after that, R01 type of grants. So I was doing pretty well. I mean, at the early stage of my career. So.
What were the R01 grants for? What's the research at that point?
The R01 grant was on hepatitis B virus. and looking at some of the variants, some of the functions and how they impact disease. And so that was what the grant was about. That grant was funded by NCI, that one grant, because I kind of twisted to more of a cancer. And NCI obviously has interest in hepatitis viruses because it's an oncogenic virus, right? So they have funded work related to liver cancer, and they continue to do so, which is fine. I mean, NIH is sort of, it really depends on where you can get the money, right? NIH.
Right.
So, yeah.
Right. So were you aware of Jay Hoofnagel's work? I would assume you met with him and things like that.
Yeah, I mean, actually, he's the one sort of approached me when I was at Harvard Medical School about looking at this particular job, liver disease. Back then was the sections. And I really didn't know a lot about intramural program of NIH. I never thought about it. I mean, for me, my career path would be to be moving along in the academic ranks of academic health center, right? Be a professor, maybe division chief, whatever. Obviously, the eventual hopes to be a dean or something like that. So that was kind of always my aim, and that's what kind of I have seen a lot of my mentors did. Right. So from that point of view, I didn't think much about it, but I came down anyway. I met with them, and that was interesting discussion along. I was really learning about intramural program. And I think back then there was a really a strong effort trying to rejuvenate the intramural program of NIH, because a lot of them sort of have sort of the weighing. Obviously, has always been sort of the strength of NIH. But that was because of Vietnam War, right? The Yellow Berets, a lot of the very famous people actually went through NIH at that time, and because of the war. And so that's where the name, but then obviously a lot of them left, right? And with ensuing 20 years or so. So I think there was a real effort trying to rejuvenate the intramural program to make it to be as good as it was. And I think there was a lot of so it's like a confluence of events that people were really excited about bringing people like me from elsewhere and highly promising and has a really great research track record to build the program. So I'm sort of a part of that. I guess that sort of recruitment effort back in the late 90s. So that's kind of and they were really making very generous offer, trying to do what they can, and then they have NIH has modified some of the salary structures, which make it more attractive, and in a way, I just felt that, well, I mean, why not? I mean, doesn't mean I'm going to stay at NIH forever, and I didn't think I was going to stay here forever anyway. So I thought maybe spend 5-6 years, whatever, and think moving on, and that was my thought then. And one of my mentors actually did give me a very good advice. He said that there are many of these jobs in terms of division directors in academic institution section, but there's only one job in NIH that you can make a difference at, which was a good advice. And I thought, yeah, that will give me some unique experiences. So that's what I kind of did. So I came here, and obviously I met some really, really exciting people. You mentioned there was something about Harvey Alter. Yes, that was another reason I came here. I met with him when I came down. He was just such a down-to-earth, a very thoughtful, very friendly, very accessible person. I immediately developed kinship with him. I knew of him, but I didn't know him that well personally. So that was another reason I did. I also met a few other people who work on viral hepatitis, and Bob Purcell, Steve Feinstones. Those are the people that I really kind of helped me to build this program. And I've interacted with them on and on for the years I was here. So I would say, in a way, Harvey Alter was kind of my NIH mentor, scientific mentor, because he sort of helped me. We worked together on a lot of things. And Jay also as well, Jay Hoofnagel. And so from that point of view, we really had a critical mass of people who really think alike, who wanted to do the same thing, and very collaborative from that pen. So I think that was really a great time to be here. Because of FIAU, it was a drug for hepatitis B, was testing them in the clinical center, this particular drug. And then it turned out that many patients die because the severe toxicity of this drug. That had put a lot of negative spotlight on NIH. So in a way, that section really had gone and people have left and all the things. So it was really very few people. So back then, he was only one still in the section. And he actually took on the extramural job even at that time, but he was maintaining the intramural program. So there wasn't anybody there. So that was one reason that he, I guess not just him, but the NIH leadership, those people like Alan Spiegel, who was the scientific director, Phil Gordon, who was IC director, really want to kind of rebuild this liver disease program. The confluence events really give me all the opportunities I need to do this. And so the goal was really to rebuild this program. And I think it was good that they kind of look from the point of view of they should recruit somebody who really is more of a physician scientist, really has a scientific background, strength, and expertise to build the basic science part to complement the clinical program. All right. So from that point of view, I think that's what they thought I would fit right in. And because before me, most of the research was more clinical research. I mean, right. Jay Hufnagel done some incredible research on use of interferon, treat hepatitis, and but that's all patient-oriented, which is great. I mean, that's important things. But I think the thought was really to be able to complement, have a sort of a real translational program, right? I mean, that's a buzzword. So yeah. I think that was a vision, that was a hope, and I certainly agree with that. And that's something that I wanted to do, and was good for me because although there wasn't anybody there, I had to build from ground up. But I feel that's really the opportunity I can take it however I see fit. And there again, I mean, nobody told me what I should do. They just say this is what they hope I should do, and I certainly agree with them. And then I just went ahead, did what I want to do, who I need to recruit, how I want to build, whatever. And I did not run into any roadblocks at all. I mean, people in the senior leadership was very open, very supportive, really give me a lot of flexibility and leeway to what I do, to give me the resource. So, really took many, many years to do that. And so that's where we are today.
Let's talk about hiring. There's two ways you can look at this, and I'm sure it's not as simple as that. But, one, you can envision the work you want done and hire somebody to fill that, fit in that hole. The other is you can just look for good people and bring them in and let them run. How did it work out for you?
It worked out great. Actually, a little bit of both, right? I mean, I want to hire the best people, and also I do have some programmatic interests, right? So when I advertise, that's how I would do. This is what we want to develop programs so people interested should apply. And so we were very direct in what we're trying to recruit. So which narrowed down the field in the sense we just don't recruit anybody who does great science, which is fine. I mean, that's what the philosophy of some other program. But for us, we have a focus. So that's what we did. And we got some great applicants and the candidates, and I recruited two really very promising, very accomplished women's physician scientists who was sort of their early stage, and they sort of became my first recruits. And they've been very productive, very accomplished over the ensuing 20 years when they were in my group, and they all done very well and highly recognized leaders in the field. So from that point of view, I'm sort of proud that I was able to recruit. My first two recruit have developed into a really powerhouse in the area of science. And again, they both are physician scientists, right? They are MD, but they work similarly as I do, right? That translational research, and so that's really my first recruits. And after that, because of the interest in the institute and expanding further, a lot of liver disease which have come to light, such as the obesity issue that related to fatty liver disease. So we want to develop that. There's also a lot of liver disease too. So I recruited a few of these folks from within, really from our own fellowship, from our ranks, because I think sometimes that may be the best way to recruit. If you identify the right people internally things worked out much easier because they know the system that can do this, and they all have done very well. And actually, one of them was tenure a few years ago. One of them is up for tenure, probably will be, and one was just sort of presented to the Board Scientific Council and got great, glorious marks that likely be tenure. So I'm really trying to build sort of with some focused programmatic theme, and then try to find the best people. That's sort of my philosophy of recruitment.
Okay. And as you say, there's lots of different aspects to liver disease, but two big tracks are hepatitis B and hepatitis C. Can we talk a little about how the hepatitis B work unfolded over the last dozen or more years? What were some of the landmarks and some of the big challenges in that research?
Yeah, I mean, I would say first of the discovery of hepatitis B by Baruch Blumberg. I think almost 50 years ago, that sort of led to his Nobel Prize, was a major discovery. And Harvey Alter actually was involved in that, right? Although he didn't get the Nobel Prize for that, but he was very much involved in the initial discovery of hepatitis B. He works for Baruch Blumberg, but he certainly made major contribution. So that was the first thing. And then quickly after that, actually the vaccine was developed just really by taking HBV infected patients' serum, isolate their viral antigens and activate it, and it becomes a very effective vaccine. So obviously they undergo various different improvement process of the vaccine. And it's up to date still, hepatitis B vaccine is probably one of the most effective vaccines. And also as a fact that it's a cancer vaccine, right? Because you prevent HBV, which can cause cancer. So that was one of the major milestone discovery. So when I came into the field, that's what we have. Right. But there's a lot of interest in obviously therapy. Even when we have a vaccine, there's still a lot of people, million people infected with the virus. They can develop cancer, develop end-stage liver disease, die of cirrhosis. So there was a lot of interest in therapy. And so from that point of view, really sort of the history rolled out in the sense that obviously develop therapy, we gotta have good model systems to study the virus, cell culture, animal models, and all these things that are sort of unfolded over the 20 years. We have certainly learned a lot about the virus. And then during the meantime, that we were able to develop a drug that can suppress HBV infection, not necessarily cure at this point. That's kind of state of art in therapy. And much of this actually benefit from a major investment in the HIV field, right? Because a lot of drugs are similar, sort of gotten tested, not in HIV, but tested in HPV. So it's sort of benefited from that. So we actually, right now, we do have a very effective drug in suppressing viral infection, just like in HIV, right? You need to take that. Unfortunately, you have to take that for perpetuity, because if you don't, your virus can come back. So that's the same state of knowledge we have right now with hepatitis B virus. So the next goal is really trying to develop curative therapy, right? Instead of putting somebody on for an indefinite period of time, hopefully you can come up with something that can cure the viral infection. It's a tall order, but I think it's doable. And that's a similar strategy that people in the HIV field are pursuing, trying to develop a curative therapy. So that's the state of science in HPV. So we've been involved in various different steps and stuff. So.
This is an interesting point. HIV has come up a couple of times here. Just talk a little bit about how HIV and HBV are related. Why is it that they pose similar challenges?
Well, first of all, I think from the public health epidemiology point of view is that they share a lot of common routes of transmission. But also between HPV and HIV, actually, they are sort of ancestrally related viruses. People may not know this. I mean, that's sort of been the case, and has a very similar structural organizations and some of the functions by the virus. Again, it's a it's a reverse transcriptase type of a process as well. So the HIV virus required to replicate, so is the HPV virus. So that's what I was saying, that some of the drugs that develop against HIV targeting the reverse transcriptase turn out to be active against HPV reverse transcriptase. So that's where the transition happened. So absolutely, they are common, and there are people are co-infected with the virus, two viruses, and that's actually not uncommon that we see. So there's still a lot of close resemblance between two viruses. So,
you mentioned the modeling,
yeah.
and one of the things your lab did was develop some new ways of modeling, I guess. Talk about
HCV. Okay, we haven't really talked much about HCV, but
yeah.
well, we did, but not in terms of the science. So,
well, is there anything about HBV that we should cover before we move over?
Well, I think right now, the primary goal for the scientific community in the HBV field is to develop curative therapies. I would say that's, we have a pretty effective vaccine, right? And then the suppressive regimen works pretty well. So if we can make sure that we have a curative therapy, we can really eliminate that as a public health problem. And obviously, there's a lot of issue in terms of public health measure in terms of implementing universal vaccinations and testing, all the stuff like that. That's sort of been adopted actually for HPV, universal vaccinations and testing. So I think a lot of it just have to take advantage of what we have to apply to the masses, and that could control the infections. Yeah.
Well, let's move over to hepatitis C then and talk about that modeling challenge.
Yeah, again, when we first know about the virus, and there weren't a lot of tools to study this, and really took a while for the field to develop. First of all, some kind of cell culture system, and initially it was just basically a forced expression of viral gene in cell cultures, not really a natural infections model. And we were involved in sort of the demonstration of a first infectious cell culture system using this particular strain of virus that was isolated from Wakita's lab back in Japan. And that was just really one of the greatest collaboration I have. We worked together on that. We generate very efficient cell culture system using the viral strain that he had uncovered from the patient with hepatitis C. And then that led to a whole series of discoveries and modifications. So now we actually have very good infectious cell culture system of various different strains or genotype of virus, so we can really look at them, right? Because obviously, if we want to develop therapy or vaccine, they need to be effective against all the different strains and genotypes. So that's why it gives us an opportunity to do that. So that's really been one of the greatest advances in the HCV field, developing all these model systems. And again, with the cell culture, with the cell culture system, it's not sufficient to develop vaccine, right? So vaccine, you need an animal model. So that's why chimps become such a critical tool in developing vaccine against human viruses, especially hepatitis viruses, because that's not like HIV. HIV has sort of the monkey model, which kind of works pretty well. That's why they don't really use chimps. But in HBV and HCV, that's not the case. We did really a good monkey model for the viral infections because it's much more limited. So therefore, chimp become critical, and we still have a long way to go because vaccine still, without an animal model, is very difficult to test your vaccine candidate unless you go straight to clinical trials, which had happened. Yeah. So go ahead.
I just want to unpack the tissue culture and talk about the process. Why was it that HCV didn't work with the more traditional techniques, the things that everybody was doing at the time? And how did you develop a relationship with Wakita and get this development made?
Yeah, that's probably true with most of the hepatitis viruses. They're very finicky. They it's not so easy. It's not like some of the viruses, you can just throw them on the cell culture. They will replicate, they will infect. It seems to be not the case. People certainly have tried. It's not for lack of trying. People have tried all kinds of different cell culture systems. None of them seem to work because maybe evolutionally that's what the virus evolved to be. Not sort of doesn't really replicate that robustly because what exactly does in terms of causing chronic infections as opposed to acute infection with most of the viruses, right? So it really take a while for people just by trial and error to identify potential cell culture system, and also using something more of a primary cell culture system like the primary human hepatocyte instead of using transformed cells, which are most of the cell culture system, right? They are cancerous cells. And there's probably a reason why the virus doesn't replicate well in cancerous cells because they like to infect the physiologically normal cells. And so it took a while to figure it out, and certainly the identification of the strain. And that's actually where it came about, because Wakita was a postdoc when I was at MGH. All right. So we kind of developed that relationship. And after we, I moved here, he went back to Japan. We continued to collaborate, work together. And and then one day he reached out to me. He says, "Hey, we have this patient with fulminant hepatitis C, and we want to sequence it, see why is that the case, and would you work together?" And then we kind of did, and that led to the history that apparently the strain that he initially uncovered from the patient turned out to be very useful cell culture model that can replicate very well in the cell culture we have. I also should take a pause, say that before that, there was this development of recounts, right? You kind of, by Ralph Bodenschlager and Charlie Rice, that they kind of was able to introduce a viral genome into the cells and will replicate, but it doesn't produce infectious virus. So it's not infectious cells, but that's still very useful, right? Because you can have a replication system, although it's not infections, but you still can look at how the virus replicates, right? So that was certainly another milestone development, hepatitis C. And then also people started playing around with different animal models, right? With this humanized chimeric mouse models that be able to grab primary human hepatocytes into the immune compromised mice that can be infected by hepatitis C virus. And as a matter of fact, can be used for other hepatitis viruses as well, including hepatitis B. So that sort of led to opportunity to look at sort of in vivo models. But again, they are not the same as chimp. Chimp is still necessary for vaccine development because you're dealing with immune response, whereas in these humanized mouse model, you don't have the immune system to study. So that's, I think, is there anything particular other area you want me to expand on? Or
we really want to get to vaccines, and I can see why. I just want to make sure we wrapped up the modeling thing. Wakita, the cell culture that he found, or the tissue culture that he found, was this just the patient just happened to have these particular cells, or how did that work?
No, actually it's not a cell from the patient, it's a virus from the patients. And actually the interesting connection was this Japanese patient, developed fulminant hepatitis C, which is very rare. Fulminant means that you develop liver failure. Usually we don't see that. Usually people infect the hepatitis virus has a very mild disease. Maybe they're a little sick, like flu symptoms, but they really develop liver failure. But it does happen to call acute liver failure. And what's actually more interesting in some of my earlier work when I was a fellows, I actually study fulminant hepatitis B. All right, so it's it's a B, but C. We see that in both viruses, right? So that's actually, I did some of my earlier work on hepatitis B, looking at these fulminant hepatitis B patients, and identify actually a very interesting mutational signature in these fulminant hepatitis B patients. So that's really kind of my initial claim to fame. All right, when hepatitis B. So that kind of become logical. Sort of, he thought, hey, I worked on hepatitis B, the fulminant disease. There's something commonality, and let's look at this together. I say, oh yeah, of course. So we began to work together. So it turned out really the virus strain that he uncovered in this patient turned out to be very efficient in infecting and replicating in cell cultures.
Okay. And so the implications of that for hepatitis B then moving forward, what was that contribution of that specific technology that you
hepatitis C? So, it has enormous, because now we can grow the virus, we can do reverse genetic, forward genetics, we can mutate the virus, we can look at the effect of certain mutations, looking at strains and genotypes, and have a very convenient cell culture system, just like other virology, right? I mean, virology benefited being by bioavailability of a lot of cell culture systems. So if we can grow them, study them, mutate them using reverse genetics. And this is very much also went along the line of what we face currently with the COVID, right? Without cell culture model, it's very difficult to study. Fortunately, with the SARS virus, that it can infect a lot of cells. You don't have to do much search and probe, and we have that. And actually, that's sort of critical, important in a lot of the studies that people are doing now, especially with antiviral development, right? With cell culture. That's sort of the key for antiviral development. Whereas animal model would be the key for vaccine, right? So, yeah.
Okay. So one of the things you talk about, there are treatments, but we still need a vaccine. Talk about the interferon nucleoside reverse transcriptase inhibitor therapy, and how that sort of has created sort of taken the pressure off of the drive to
vaccine. Yeah, I mean, I think certainly vaccine helped, but there are million people infected with the virus, so vaccine is not going to help those people, right? And also, people continue to be infected even though there's a vaccine, because there has not been sort of a universal vaccination program in the world in terms of protecting the most vulnerable population, which is the sort of the developing world, right? They don't have the resource nor the opportunity to mass vaccinate their people, although it's done pretty well in some part of the world in terms of HPV vaccine. So I think from that point of view that vaccine is not going to do it. And also there are people dying from hepatitis B, and vaccine is not going to help, right? So the only way to do that is to treat them. And that's why I refer to the sort of the knowledge we learn in the HIV field is able to apply to the HPV with this NRTI, the reverse transcriptase inhibitors, and that certainly has major impact in terms of slowing or controlling the hepatitis B virus, so people don't develop cirrhosis and liver cancer. And that's been shown over and over again, large population-based study, that these drugs work. I mean, you mentioned about interferon. Interferon was sort of initially the drug that being used to treat hepatitis B. It's advantage and disadvantage, and obviously it's not as easy because it's injections with this reverse transcriptase inhibitor. It's pills, which is always easier. And so from that point of view, yes, it has an impact on the reducing the disease burden of people who are infected with the virus, whereas vaccine would prevent new infections. And so both have a unique complementary approach. So I think that's why it's so important to have a strategy to develop both way of controlling the viral infections.
And let's talk about some of those vaccine strategies. I was interested to see that you'd done some work with an adenovirus vector.
Yeah, I mean, adenovirus seems now has become sort of a well, not totally the vector choice. It's only been a very popular vehicle for vaccine development, right? And, in terms of the SARS, I mean the Johnson & Johnson's adenovirus based SARS vaccine. So obviously the RNA based vaccine is a new development that would be interesting. Whether it can be applicable to HCV vaccine remain to be seen. But so adenovirus has been around, and we've been using that not only to express genes because they're very efficient. The adenovirus will infect cell culture very efficiently, so you can really use that as a transfer vehicle to express gene of interest in a foreign gene of interest in the cell, and that's only been used also as a vaccine candidate, and that's been tested, for many years in various different infectious process. So we did some work related to that, but I would say that actually we didn't really pursue that avenue using adenovirus as a vaccine candidate. We were focusing on sort of a technology we developed in the lab by expressing the co-protein of the virus in an insect cell system. And lo and behold, they're able to generate virus-like particles. All right, they look like virus. If you look on the EM, we've done a lot of work related to that, and then we thought, hey, that could be a good vaccine candidate, right? Because it's not infectious; it just has the coprotein. It looks like a virus. So we actually done a lot of work related to that, trying to show that it can induce immunity, and we've done that in the cell culture and in the animal model. Eventually, actually, we even did a study on chimps, and that's what was so important to show that indeed the vaccine candidate, this virus-like particles, can induce immunity enough to prevent viral infections and chronicity of the hepatitis C virus. So that was sort of our last work. That was many, many years ago. It became difficult to move forward because, first of all, the chimp moratorium and pharmaceutical company was so focused on therapy back then, for the last 10, 15 years to develop therapy instead of vaccines. So couldn't get a lot of interest elsewhere to help us do this. I may get back to it now. But anyway, so we have not really tried the adenovirus. And I think you're referring to the adenovirus was the one that the GSK or chiral vaccine candidate who was tested by NIAID and published in New England Journal of Medicine last year. Unfortunately, it didn't work, right? But that sort of illustrates the difficulty of developing HCV vaccine. It really took them 10 years to look at this and millions of dollars, and turned out to have a negative trial. Although we did learn a lot from that trial, even though it's a negative study. All right, so that's science, right? So I think from that point of view, it's really illustrated the difficulty of developing HCV vaccine without a convenient animal model system, without infrastructure support, without some way to facilitate the pipeline. So that's really been the fundamental problem why we don't have HCV vaccine. Whereas with hepatitis B, I mean, I mentioned to you, right? A few years after the discovery of hepatitis B, we had a hepatitis B vaccines. And so.
Now, were you going full bore on the chimpanzee work?
You mean, do you mean the chimpanzee or the control human infection model? The chimp.
Yeah. I'm thinking about the chimpanzee work before the moratorium.
Oh yeah, we were, but then that basically put a stop on it, and we also have pivoted to some. To therapy, actually, we did that too. We pivoted to therapy, and
as the drug companies did, same.
Just because it was easier, and we get fundings and all the things like that. So
and the chimeric mouse models was another one.
Yeah, that too. We can test in vivo. We've done that with some of our compound that we have developed in our laboratories, and so we have tested some of those. And unfortunately, with some of the compound that we have developed in our lab, probably not going to really make into the masses because the pharmaceutical company have been much more successful in developing effective anti-HCV therapy. So I'm not sure whether we'd be able to compete against them. And so anyway, I mean, it is what it is. So we kind of we'll continue to work on that, but we are probably going to pivot more back to the vaccine. This is sort of get to the point. And I thought the way to get back to the vaccine, knowing that I've been involved in the early stage of vaccine development, HCV vaccine development, the moratorium of the chimpanzee, and then the failure of the recent trials, sort of gave me pause in terms of yes, we can do all these basic science work, but where is going to go once you get there? It's really a major roadblock, right? So that's what sort of got me to thinking about this so-called control human infection model, which maybe that's what you were referring to. And this has actually been around for decades, not just hepatitis C, but in other viruses as well, to use that as a platform to facilitate, expedite testing of vaccine candidate in humans in a very controlled systems. Obviously, a lot of ethical, regulatory issues with that. I'm not going to go into that. I wrote an opinion piece or perspective in New England Journal last year, and that sort of addressed some of the issues. But I think without something like this, unless we resuscitate the chimps effort, I don't think we're going to get a vaccine. That's my personal feeling. And I think that's the same with people in the field, that we need some way to move forward. And therefore, I think the control human infection model is a unique way to look at that. I don't know whether we're going to get it off the ground, but I think the stars are aligning. I think people are realizing that we got to be thinking outside the box to do some of the stuff. And so that this is sort of actually because of the COVID that this actually was revived, right? These human challenging study with COVID to study the vaccine or antiviral sort of, et cetera, and so on. Fortunately, we didn't really need those model for the vaccine for COVID. But still, I think sort of the same concept. There's been a lot of interest in discussing this whole potential of control human infection model.
So you've done a lot of thinking about this, the CHIM approach, I guess. Yeah. Where has that worked? What are some examples of successes for that approach?
Oh yeah, I mean, it's been tested. Actually, the first one was tested was in diarrhea, in infectious diarrhea. Was cholera. Yep, actually, the first study of cholera, people intentionally give people cholera. I mean, give them vaccine, give them cholera, and see whether they develop symptoms. That's actually, it was 20, 30 years ago. That was the first example, control human infection model. And subsequent to that, I would say malaria, then dengue virus has been tested using fairly successfully in the control human infection models, and influenza. That again, that's another area that people have been using to this model to sort of intentionally infect somebody in a control setting and then see how they behave if you give them vaccines. And there's some example of success. I'm not saying this is applicable to all the infections, but I think for some of them, certainly for hepatitis C, I think the science has evolved to the stage that would be able to use it because we have very effective therapy now. I mean, that's a game changer, right? Because in the control human infection model, you're always concerned about what's a consequence. If somebody develops, infection, if you can't treat it, they can develop complications, and right? I mean, that's what we all worry about. But if you have very effective therapies, that we can cure these people. We know that. We can cure 90, more than 95%, 99%, especially healthy people. So, and from that point of view, I think the time is here now to think about that.
Talk about some of your relationships with the other institutes. I think you mentioned NIAID, for example.
I certainly have collaborated a lot with the intramural program at NIAID, people like Bob Purcell, Patricia Fossey, and Steve Feinstone, who was at NIAID and was at FDA. And so we really had a lot of collaboration, though people interested in viral hepatitis. We actually have a pretty tightly knit group, and we kind of work together, work on different things. And I sort of, obviously, some of these key people retire, so it put a little damper on the collaborations. And but I certainly hope that we can continue to do that because there's still a lot to do in the viral hepatitis field. And so that's kind of the extent of collaboration. I've been sort of advising some of the extramural program in NIDDK. Right? They have a large portfolio in supporting grants for viral hepatitis. I have sort of served as an advisory role for them, and just sort of give so that's been very helpful. I mean, I've done that with NIDDK's extramural program people as well. That I work very closely with Jay Hufnagel, who kind of runs liver portfolios, and so we work well together. I advise on some of the new initiatives, and so that's kind of where we are, and it's been great.
And talking about sort of expanding your portfolio, you also have a role in the training program.
Mm-hmm.
Talk about what's your philosophy on training? How do you do that?
Yeah, I mean, I've been the beneficiary of strong mentoring. I mean, I would give credit to a lot of my former mentors. So I certainly think that's critical. I mean, without them, I would probably not be where I am today. So I think in a way, using my personal history and story, I can see how important it is. So my philosophy is that very often you are remembered or recognized not for the science you do, but for the people you train.
And tell me a little bit about some of the people that have made a difference that you've worked with.
Well, I think example would be the people I recruited to my branch. I mean, they all sort of very prominent figure in their own right. But in that sense, I do consider them not necessarily my trainees, but certainly the people I mentor. And when they came through, and my philosophy again is with them is that I know they need to be independent, right? They need to have their own niche. So I always try to think some way that to separate them from what I do, right? To encourage them to do something else, to focus on something, to do something different from what I do. And I will support you whatever I can. I'll advise you. I can help you. But you need to develop that kind of independence, and which is critical for anybody's career development. I think that's probably the most important transition from people from the training to a principal investigators. So I always keep that in mind, and I try to help them. I would kind of not interfere with what they do, or not to kind of always want to be part of what they do, and but in the meantime, I will support them from the distance. And so I think that's kind of very important for mentor to be. So that's kind of sort of been my philosophy. And actually, I'm sort of most proud of the fact that sometimes, like when I have somebody who's a trainee who's ambitious, who want to do well, who kind of want to do something differently, I will think about it, give them some ideas about when you focus on this, do this is whatever, I mean, run with it. I mean, I don't need to be part of it. Just I'll support you, but I don't need to be part of it. And I think from that point of view, they appreciate that. They know that you have the best interests at heart, right? I mean, I'm doing this not because I want to be a co-author on my paper. I want to take credit for what they do, right? So with transitions, you got to give people that flexibility. Yeah, I mean, certainly when I have a lot of trainees, I mean, I have to supervise them, I have to direct them, I have to do a lot of work to advance the project, which is fine. That's what I do. But I think once they started separating themselves all the time to do that, we need to keep in a perspective on that.
Another one of your roles is as deputy director of the translational research program. Everything's translational these days.
Yes.
Was that term in use when you started your career, or did it come up somewhere along the way?
No, actually, it wasn't. I would say it became fashionable about 10 years ago. And then that's when I kind of was given a nominal title to just really help with transition. I mean, it really doesn't really have any really sort of professional designations in terms of NIH jobs type of things like that. It's just sort of a title which kind of helped me to work with people. I mean, obviously, I tried to do what I can and try to integrate a lot of the basics with clinical science. And I mean, as you said, like everybody's doing that now. So whether you really need something like that, I don't know. I would say I really haven't done much in the last few years. So so the title probably meaningless at this point. So, but anyway, that was the whole idea back then.
Yeah. Or it's a measure of success that you don't need to worry about it now.
Yeah.
So where's you talk about the fact that you're kind of still building the lab and the lab's changing. Where's it going? Where do you think things are going in the future? How is the lab going to look different, say 5-10 years from now?
Well, I would say we'll continue on the theme of what we do, right? I mean, that's always been a sort of important thing for me. I mean, I could digress a bit, take some detour, but the themes always have to be there. So the theme is really sort of looking at the interaction between clinical and basic science regarding viral hepatitis and the consequence of viral hepatitis, liver disease, cancer. So there's always been a theme. So obviously the science evolved, right? Technology mature and also being discovered, and we've been pretty proactive in terms of taking advantage of some of the newer technology, such as gene therapy, such as CRISPR-Cas editing gene, and there's a lot of opportunity in the liver disease to consider some kind of gene therapy using genetic editing technology. We've been very much involved in trying to take advantage of regenerative medicine technology, like the stem cells, induce pluripotent stem cells, and et cetera, trying to use it to study viruses. So we've been kind of trying to be proactive in taking advantage of some of the newer technology to apply to our science, and it's great. And then my philosophy is always, I'm not happy doing the same things year in and year out. I want to kind of look at something new, some innovative technology, either technology or approach or platforms and stuff. And one thing actually I got very much involved is working with NCAS. They have a lot of these, it's kind of like mini drug company, right? They do a lot of drug screening, and that's kind of a lot of what we do. We kind of got involved with them, work with them. And actually I learned a lot about drug development, over the years. So which has been great. I really enjoyed that interactions. And so that's something we were proactive and kind of got into that. I think what the future holds, I think we'll continue to do a lot of stuff, and we ask questions, and we trying to look at what some of the knowledge gap and unmet needs in the area of hepatitis virus, more B and C, right? But B is curative therapy. And we need to kind of figure out some way, and maybe even better animal model in hepatitis B. Hepatitis C, I would say vaccine. It's a sort of the next frontier. So I think we're going to kind of start doing more of that, and hopefully we can bring this human infection model into place. Don't know how long it's going to take, but certainly I'm hoping that will be one way to facilitate HCV vaccine development.
Okay, now that's from your perspective in the lab. How do you think hepatitis treatment is going to look to patients?
Yeah, well, I think right now for hepatitis C, actually for hepatitis B too, but it's not curative therapy for hepatitis B, right? For hepatitis C, actually now they can take one pill a day for 8 to 12 weeks. And they can be cured. So I think that's a model. Actually, I'm hoping to be able to even do shorter, like taking antibiotics, bacterial infection, take 10 to 14 days and you're done with it instead of that long period of time, right? So I'm hoping that may becomes reality. We can shorten the course of treatments, also reduce the cost, so we can use to treat everybody. I think that's really getting to be the point that we probably should treat everybody with hepatitis C. And because before there used to be restrictions, right? Even now there are restrictions in terms of who can be treated by insurance and states and stuff like that. So my sense is that if we have such effective drugs, if it's not very expensive, just like antibiotics, we treat everybody who's in infections. So that's what I hope to see with hepatitis C. With hepatitis B, yes, we have very good drugs, but you have to take it indefinitely, right? So I'm hoping that the same thing, one pill a day for whatever, 4 weeks, whatever we can get it down to. And that's what I'm hoping. And I think it's certainly not unimaginable we can get there. I think for hepatitis C, we probably can get there sooner because we have already have very effective curative drugs, whereas hepatitis B, not so much. But it will take time. But I'm optimistic that we will get there with B. It's probably a little more difficult with B than C. For reasons I'm not going to go into. It's just a lot of reasons why I think it's more difficult to have curative therapy for B than C. One of the biggest reasons is probably because the virus has this latent form. It's like HIV, right? Why don't we have a curative therapy for HIV? Because it can hide in certain systems. It doesn't, which is hepatitis C. It's not so much. Once you hit it, then you can eliminate it. But for hepatitis B, it's like HIV. You can suppress it, but you can't get rid of it. So
interesting. Well, is there anything else that we should talk about that we haven't touched on about your career or your experiences at NIDDK?
No, I would say, I know there was some question about what I consider, why did I stay here for so long? I would say the intramural program in NIH is so unique in the sense I just love what I do. I mean, I can really quickly pivot to do what I do. I think I really give you plenty of examples of what I've done over the last 20 years, and I can shift. I mean, as I mentioned to you, actually, we're doing some work related to COVID now. So, I mean, figure that out. But it's a virus, right? So I think from that point of view, I would say that's really the greatest strength. That is give you the flexibility of people who are innovative, creative. You can take full advantage of it, and yeah, I mean, it's true that not all your study will succeed, but the key is that you gotta try hard. You gotta try early. If you fail early, that's fine. You move on. You learn from your failure. I think that's the important thing too. You can fail many times, but if you learn from it, you can make it better. And I think that's what the intramural program will allow us to do. And so I think that's what's very important about the intramural program in NIH. It's very unique in that sense. And we don't have to worry about writing grants and funding, all these things. That's really a major advantage. We can quickly pivot what we do and to do something that seems exciting and think outside the box and try something high risk, but high yield type of research.